Hello students, welcome to the biology class. Today we are continuing with our ART, that is assisted reproductive techniques. Yesterday we discussed in vitro fertilization, means the fusion of gametes outside the female body with the simulated conditions. Today we will discuss in vivo fertilization, that is the fusion of gametes within the female body, same as that is the natural fertilization. But in ART, it is being used in the females who cannot conceive, embryo formed by fusion of gametes in another female and then transferred. So difference between in vitro and in vivo fertilization. In vitro, the fusion of gametes outside the female body in the lab with the simulated conditions, whereas in vivo fertilization, the fusions of gamete within the female body. Next is gamete intrafallopian transfer. It is the transfer of an ovum collected from a donor into the fallopian tube of another female who cannot produce one, but can provide suitable environment for fertilization and further development. So it is a technique in which the ovum is collected from the donor, transferred into the fallopian tube, who cannot produce ovum but can provide suitable environment for the fertilization and the further development of embryo. Next is Intracytoplasmic sperm injection, ICSI. In this method, the sperm is directly injected into the ovum to form embryo in laboratory, and then embryo transfer into the female body to carry out for the development of embryo. Next is artificial insemination AI. This technique is used in case where the male is unable to inseminate sperms in the female reproductive tract or due to a very low sperm counts in the ejaculation. In this method, semen collected either from the husband or the healthy donor is artificially introduced into the vagina or into the uterus for the further development of embryo, that is intrauterine insemination or AUI. These all techniques are known as ART, that is assisted reproductive techniques, that is classified into the in vitro and in vivo fertilization. In vitro test tube baby program, in which we discussed zygote intrafallopian transfer, intrauterine transfer, and in vivo fertilization we discuss gamete intrafallopian transfer, that is GIFT, intracytoplasmic sperm injection, that is ICSI, and AI, that is artificial insemination. Our next is amniocentesis. It is prenatal diagnostic technique in which a sample of amniotic fluid from the pregnant woman is taken during the early stages of fetal development and cells are cultured and analyzed. Advantage of this method is, by this method, chromosomal abnormalities, sex of the fetus and the developmental disorders could be detected for treatment. But instead of using as the diagnostic technique, it is misused for killing the normal female fetus, and there is a ban on this technique to legally check female feticides. So this particular diagnostic technique, earlier it was used for the chromosomal abnormalities, but due to the sex determination, the killing of the normal female feticides is increasing day by day. This technique is being banned to legally check the female feticides. Next, we are having some Cbse important questions. As we have already discussed that in section A, we will have a multiple choice question. Section B, that is 2 marks question, that is very short answer type questions. Section C will having a 3-mark question. Section D, the figure based question, and section E is long answer type question of 5 marks. This is all about this chapter. So we have finished with the reproductive health. Go through this chapter through NCERT. Try to solve all the in-text as well as the exercise question. On Monday, I will take your doubt class on Zoom app at 4 p.m. Till then, goodbye. Thank you.